What if I told you that your morning coffee could be a secret weapon against infections? Or how about a milk that stays fresh for months rather than spoiling in just days? The global demand for sustainable and healthy animal-based food products is on the rise, with an expected 70% increase by 2050. To meet a demand so substantial, we need to turn to advanced technology, genetically modified organisms. But would you be surprised if I told you the future of milk could lie in the hands, or should I say hooves, of transgenic goats? Welcome to the genetic basis of stuff and things. Genetically modified organisms have played a role in the development of some incredible advancements since the 1970s. From insulin-producing bacteria to pest-resistant crops, altering the genetic makeup of organisms has revolutionized numerous industries. In fact, the livestock industry has long been a focal point for genetic innovation. In 1985, researchers successfully engineered transgenic rabbits, sheep, and pigs via microinjection of foreign DNA. Turns out it wasn't just the stuff of mad scientists in movies. Since then, the focus has shifted towards the dairy industry, enhancing milk's natural properties like introducing antimicrobial benefits. This led to transgenic cattle being used in groundbreaking microbial research, especially involving lysozyme, an antimicrobial enzyme highly expressed in human breast milk, tears, and saliva, and mucus. Believe it or not, the discovery of this major enzyme was a complete accident. In 1922, Nobel Prize-winning physician and microbiologist Alexander Fleming, the same man who discovered penicillin, accidentally dripped some of his mucus onto a petri dish full of bacteria. Whilst numerous colonies of bacteria proliferated, the area where the mucus had been inoculated stayed clear. That was lysozyme doing its bacteria-fighting magic. As part of the innate immune system, lysozyme hydrolyzes the beta-1,4 glycosidic linkages in the peptidoglycan of the bacteria's cell wall. Fleming showed us that lysozyme helps slow the growth of harmful bacteria, sort of like a microscopic shield within our bodies. But what happens when that shield is compromised? A 1993 publication revealed a concerning link between point mutations in the lysozyme gene, or LYZ gene, and amyloidosis, the rare but fatal buildup of amyloid that leads to organ failure. It just goes to show how much we depend on the protective power of lysozyme. Now, lysozyme is naturally present in human milk at concentrations up to 3,000 times greater than livestock milk. So, what benefits will we see if we could enhance this concentration in livestock milk? The first transgenic models for human lysozyme were mice. Small, easy to transform in large numbers, but have you ever tried milking a mouse? So researchers turned to transgenic goats. Smaller than cattle, short gestation period, and not to mention absolutely adorable. Enhancing the lysozyme within these goat models led to astounding findings on udder health, particularly developing a natural resistance to mastitis. While this extremely painful inflammatory response in the mammary gland is commonly associated with pregnant women, you might be surprised to learn that this painful swelling affects goats and cattle too. In 2006, researchers at the University of California, Davis pushed their work with these super goats even further. No, I'm not talking about goats with super strength or laser vision, but these transgenic goats were one of the first, where a species had been genetically engineered to produce milk with additional beneficial properties for human consumption. I'm sure you're picturing a half goat, half human by now, so let's shift gears and dive into the genetics behind this remarkable protein engineering. The question researchers faced was simple. How do you make sure that the LYZ gene only turns on in the milk and not in other parts of the goat? The answer lies in gene regulation, which can be influenced by specific regions at the beginning and end of a gene. These include the promoter, the 5' untranslated region, and the 3' untranslated region. But which gene are we talking about here? Introducing the bovine alpha S1 casein gene. This gene plays a crucial role in casein production, an abundant protein in bovine milk. Fun fact, casein also gives milk that rich white color. Scientists use pronuclear microinjection, a technique where a transgenic DNA construct is injected into the pronucleus of a fertilized goat embryo. This DNA construct consisted of the 23 kilobase alpha S1 casein promoter sequence, which turns on the expression of the LYZ transgene in the mammary gland, along with 3' regulatory elements sequence and a 540 base pair complementary DNA sequence for human lysozyme, the gene of interest. Utilizing the alpha S1 casein promoter is effective because it is naturally active in the mammary glands during lactation, and so scientists were able to direct the expression of human lysozyme in the milk. As a result, these transgenic goats produced milk with human lysozyme levels that were a staggering 1,080 times higher than what's naturally found in regular goat's milk. This seminal publication was the beginning of a journey of some incredible advancements towards animal health and human nutrition. Since these early works, transgenic goat's milk had been shown to modulate intestinal microflora in pigs, enhancing their gut health. Researchers have also been fine-tuning the alpha S1 casein gene in goats, aiming to reduce the allergenic potential of their milk, a significant step forward in food safety and dairy tolerance. And if that wasn't enough, transgenic cows are now producing milk enriched with human lactoferrin to improve the gastrointestinal tracts of young pigs. And isn't that just legend, wait for it, dairy?